So I was not on the Today Show this morning, but I'm still going to try to follow Dr. Grubb here. So as Chip said, I now work with the clinical in vivo gene therapy group, but back in the day, I was a coordinator on the LCA Luxturna trials. So I'm going to talk a little bit about gene therapy approval, clinical trials from that side, from like a coordinator's perspective, site management perspective, which is a little bit different. So a little bit of background, like Chip was saying, Luxturna treats a type of inherited retinal dystrophy that's caused by a mutation in the RPE65 gene. It's called Leber's congenital amaurosis type 2, retinitis pigmentosa, early onset retinal dystrophy, lots of different things depending on your clinical diagnosis. But the absence of that protein leads to degeneration of the photoreceptors in the retina and can lead to total blindness. So main symptoms of the disease are nyctalopia or night blindness, a constricted visual field, and progressive loss of visual acuity. You can see here from one of our natural history studies, the progressive visual field loss in a patient, a sample patient, age 6 on the left and age 23 on the right. So leading to almost total blindness by early adulthood. So thinking about FDA approval, we think about safety and efficacy. Those are the two things that we're looking for in a clinical trial. So the FDA is looking to protect the right safety and welfare of the participants. So that goes to our Code of Federal Regulations and our good clinical practice. So we've heard a lot about GMP and GLP. We also have GCP. So good clinical practice is an internationally recognized set of standards for conducting clinical trials. So we adhere to those as well as the federal regulations set by the FDA. And the most important one, of course, is the protection of human subjects. And that centers around the informed consent. So making sure that participants are fully aware that they're participating in research. They know that there are inherent risks, both known and unknown, and that there's a potential that they won't benefit at all from participating. So the regulations are the same, whether you're trying to license the product or not, at least on the site side. The sponsor has an entirely different set of responsibilities, but the regulations and our adherence to them is especially scrutinized in a licensing trial. So strict adherence to those protocols, to those regulations, and thorough documentation is absolutely critical because the likelihood of an audit is very high, especially in these rare disease trials like LCA. We only had 2 sites for the protocol at CHOP and the University of Iowa, and we were both audited by the FDA. And they will go through your books line by line, compare every piece of data to every piece of data that was submitted to the FDA with the licensing agreement. So you want to make sure that you're super organized and that all of your documentation is in order. And then going into the efficacy portion, you can see here from an FDA draft guidance document that they actually acknowledge that for rare diseases, you might need to develop your own endpoint because there is no clinical endpoint characterized. And that's exactly what happened with Luxturna. So the study teams and the sponsor consulted with the FDA to develop the multi-luminance mobility test, or MLMT. And we designed it as a way to sort of replicate life for these patients to the best of our ability, what we call functional vision, because we wanted to be able to measure a change in independence, a change in the quality of life for these patients that can't really be captured with just an eye chart. So we conducted this, it's essentially an obstacle course. We conducted it at a bunch of different light levels to replicate the nyctalopia phenotype. So this is the basic outline of what would become the primary outcome measure for Luxturna. And there's 12 sets of these, all different orders, same number of arrows. So you have the arrows here that delineate the path that the patient is supposed to follow. There are obstacles that they step onto, like a step, and then there's obstacles that they step over, and then there are obstacles along the side of the path that are at varying heights to measure different visual fields. And then at the end is a door where you touch the doorknob to end the course. So once we designed this, of course, we had to validate the assay, which led us to the mobility test validation study. And this was a prospective observational study. We enrolled normally sighted and low vision participants. All of the low vision participants had inherited retinal dystrophies, but not all were due to RP65. So we designed the study to evaluate the construct validity, so ability to distinguish between normally sighted and low vision, the reliability of the course. So when we sent these videos off to independent graders, wanted to make sure that there was reproducibility in their scoring. Content validity, so how it correlates to other measures of vision like visual acuity, visual field, and patient-reported outcomes. And then the ability to detect change. So from baseline to 1 year was what we did in this study. So just a little bit of data here from the validation study. On the left in the top panel, you can see that the MLMT can definitely distinguish between normal sighted and low vision patients. On the top panel, all of the blue circles are passes on the mobility course. And then on panel B on the bottom are the low vision subjects who you can see have a very wide range of performance. The circles are passes and then the red X's are fails. And you see such a wide range because this includes all light levels. So participants who failed at a darker level and were able to pass at a brighter level, they're all included there. And then on the right side, you see the ability to detect change. So the red subjects are the normal sighted with no change from baseline to 1 year. And then our low vision subjects, you actually have 8 out of the 28 low vision subjects that got worse over the course of a year. 6 subjects changed by one light level, meaning that they could do the course at 1 light level brighter than at baseline. And 2 subjects actually decreased by 2 light levels. So once we establish that endpoint, that leads us to our phase 3 trial of Voretigene Naparvovec, which would eventually become Luxturna. And this was a randomized controlled open label study. It was not blinded because we wanted to minimize the risk and patient burden of doing 2 sham surgeries on kids, 2 additional sham surgeries in the control group. So it was a crossover design. The control group was followed for 1 year without intervention, and then they were assessed at the 1 year mark and compared to the intervention group at 1 year. So, and it was not blinded, as we said, but the graders were blinded. So when the video was sent to the independent reading center, they were masked as to treatment group. Oh, okay, so here's a video. On the left is baseline, and on the right is 1 year. This is one of our Luxturna participants. So, and this is the same light level. So you can see at baseline, she's feeling her way with her feet, really not sure where she's going, not confident at all. And then on the right, she breezes through, no problem at 1 year. Totally different level of confidence in just 1 year at the same level of light. So, and then here, just the data from the mobility course, the intervention subjects in green and the control subjects in blue. You can see obviously a distinct difference between the 2 in that 1 year. And 13 out of the 20 intervention subjects passed at the lowest light level we tested, which was 1 lux. So that's the maximum improvement we could even measure on mobility. And no control subjects passed at 1 lux at 1 year. So December 2017, we received FDA approval for Luxturna. It was a great Christmas present. I believe it was December 20th. So we got in there right before the holidays. And just tips from a site perspective, from a coordinator perspective, plan ahead, especially for rare diseases. As you saw, we had to develop our own outcome measure, and I think that may be the case for a lot of these upcoming rare diseases. And discuss your plans with the FDA. They're very open to having discussions about what's the best way to move forward. And then from a CRC perspective, document everything. Absolutely, more than you even think you should. And then ask for help, which brings me to the CHOP resources. At the top, a plug for our group, the Clinical in Vivo Gene Therapy Group. We provide advice, support to clinical PIs doing gene therapy throughout the institution, regardless of therapeutic area. Our CRCs are gene therapy specific specialists, so they do end-to-end trial support from startup to closeout. And we also have a translational regulatory specialist to help move products from bench to clinic. And then also at CHOP is the Office of Research Compliance. They were enormously helpful in helping us prepare for our audit. And they will help you even if you don't have an audit. They will just help you make sure that all of your documentation is in compliance. And then the biostatistics and data management core can help with biostatistics and data management. They will help with study design, randomization, statistics, and then they can design a database or even manage a database for you. Oh, I lost my acknowledgement slide, but
Here,
there it
sorry.
is.
I don't know why they
Acknowledgements. Too many people on the study teams, but I listed all the PIs here, and then of course all of our participants and families, not just Luxturna, but the validation study as well, because we could not have gotten Luxturna approved without that study. So